All right, let's get ready to dive deep today because we are tackling something you might not realize impacts you every day, and that's scientific depth.
Ooh, that's a good one.
Yeah. So we are going to be taking a close look at a set of guidelines called MIQE,
Okay.
Minimum Information for Publication of Quantitative Real-Time PCR Experiments.
I've heard of those.
Yeah, now I know that sounds like a mouthful, but stick with me.
It's actually a pretty important mouthful.
Yeah.
These guidelines are all about ensuring that research using a certain technique called qPCR is top-notch,
Right.
reliable, reproducible, and ultimately trustworthy.
Yeah, and you know what? Before we even get to MIQE, let's break down this qPCR thing.
Okay.
Why should we care about it?
Well,
What is it all about?
qPCR is like a molecular magnifying glass.
Okay.
It allows scientists to zoom in on specific genes and measure how much of them are present in a sample. It's used in so many fields, from diagnosing diseases to tracking viruses, even analyzing crime scenes.
Wow, so it's kind of like a detective tool for the microscopic world.
That's a great way to put it.
Yeah.
And the amount of a particular gene present can tell us a lot. For
Okay.
instance, it can help determine if someone has a certain genetic condition or how their body is responding to a treatment.
That makes sense. But, our source mentions inconsistent reporting of these qPCR experiments.
Right.
What's the issue there?
Well, the problem is that without clear standards, scientists might report their qPCR experiments in different ways, making it difficult to compare results from different studies or to repeat those studies accurately.
Yeah.
It'd be like trying to bake a cake where everyone has their own version of a cup or a teaspoon.
Oh.
Chaos in the kitchen.
So essentially, MIQE is trying to bring some much-needed order to this scientific baking process.
Exactly. MIQE lays out a clear set of guidelines for reporting qPCR experiments, ensuring everyone is on the same page using the same measuring cups, so to speak.
I like that analogy.
This consistency is vital for what we call analytical specificity.
Okay, analytical specificity sounds important. Break that down for me.
Think of it this way. If you're fishing, you want a net that catches the specific type of fish you're after, not everything in the ocean.
Right.
Analytical specificity ensures that the qPCR test is only detecting the target gene and not picking up other genetic material that might be present.
So it's about eliminating those false positives.
Yes.
Those red herrings in the scientific investigation.
You got it. And another crucial aspect of scientific depth, especially in a field like molecular biology, is the use of controls.
Controls. What role do they play in all of this?
Think of controls as safeguards that help validate the results of an experiment.
Okay.
One common type is the no template control, which is
Yeah.
basically running the qPCR reaction without any sample.
Interesting.
If you still get a signal, there's contamination somewhere and your results might be skewed.
So it's like making sure your equipment is properly calibrated before you take any measurements.
Exactly. It's all about minimizing the chances of error and ensuring the data you're getting is accurate and reliable. And this attention to detail is what truly defines scientific depth. It's about going beyond just getting a result. It's about understanding the nuances, the potential pitfalls, and taking every possible step to ensure the reliability of your findings.
It's like the difference between skimming a news article and really digging into the details,
Right.
examining the sources and forming your own informed opinion.
Absolutely. And just like a good journalist fact checks their sources, MIQE encourages researchers to scrutinize every aspect of their experiments. For instance, it emphasizes the importance of meticulous record keeping,
Okay.
especially when it comes to sample handling and preparation.
Why is that so crucial?
Well, even seemingly small variations in how samples are collected, stored, or processed can have a big impact on the results of a qPCR experiment.
Really?
Think of it like baking bread. If you use old yeast or let the dough rise for too long, your loaf won't turn out as expected, even if you follow the rest of the recipe perfectly.
So it's like MIQE is saying, don't just tell us what ingredients you use, tell us exactly how you handled each one.
Precisely. And MIQE goes even further, stressing the importance of controlling for things like RNA quality and integrity, especially in experiments that are looking at gene expression.
Now, why is RNA quality so important in this context?
RNA is a lot more fragile than DNA and can degrade easily.
Okay.
If you're working with degraded RNA, you're essentially working with corrupted information, which can lead to inaccurate results.
So it's like trying to read a book with missing pages or faded ink.
Yeah.
You might get the gist of the story, but you're going to miss crucial details.
That's a great analogy, and MIQE recommends using various methods to assess the quality of the RNA before using it in an experiment.
Mm.
It's like double-checking your book for any torn pages or smudged words before you start reading.
It sounds like MIQE is really setting a high bar for researchers, pushing them to be incredibly thorough in their work.
It is, and that's a good thing.
Yeah.
Because ultimately this rigor translates into more reliable and trustworthy results, MIQE even delves into the specifics of the reverse transcription step, which is where RNA is converted into cDNA, the molecule that's actually analyzed in the qPCR reaction.
What's so important about that particular step?
This is where a lot of variability can slip in.
Okay.
The type of enzyme used, the reaction conditions, even small variations in temperature can all affect how efficiently the RNA is converted into cDNA. Any inconsistencies here can throw off the entire experiment.
It seems like MIQE is leaving no room for sloppiness or cutting corners.
It's not about being a stickler for rules. It's about understanding that every step in the process matters, and controlling for those variables is essential for achieving scientific depth. MIQE also emphasizes documenting every detail of the qPCR reaction itself, like the specific primers used, the concentrations, the cycling conditions, basically the entire recipe for the experiment.
So it's not just about transparency, it's about creating a level of detail that allows other researchers to replicate the experiment and verify the findings.
Exactly. Reproducibility is a cornerstone of good science, and MIQE is all about ensuring that research is robust enough to be replicated, scrutinized, and ultimately built upon by other scientists. And of course, scientific depth also hinges on evaluating the performance of the qPCR assay itself.
Okay, let's talk about assay performance. What are some of the key factors that researchers need to consider here?
One of the most crucial is PCR efficiency. This is basically a measure of how well the target DNA is being amplified with each cycle of the reaction.
I imagine it's like the efficiency of car engine. You want to get the most mileage out of every drop of fuel.
That's a great way to think about it. A high PCR efficiency means you're getting a strong signal, and your results are likely to be more accurate. MIQE recommends using calibration curves to determine the efficiency of the assay and to ensure that it's performing consistently across a range of target concentrations.
So it's like calibrating your scientific instruments to make sure they're giving you accurate readings.
Precisely, another key factor is the assay's linear dynamic range, which is the range of target concentrations over which the assay produces reliable results.
So it's like the zoom lens on a camera. You want to be able to get a clear picture whether you're zoomed in tight or taking a wide-angle shot.
A good analogy. A wide linear dynamic range gives the assay more versatility, and equally important is the assay's limit of detection, or LOD. This is the lowest concentration of the target that the assay can reliably detect.
I'm guessing this is particularly important in situations where you're looking for something that might be present in very small amounts, like traces of a virus or a specific biomarker for a disease.
Exactly. If the target is below the LOD, you might miss it entirely. And finally, MIQE also stresses the importance of evaluating the precision of the assay.
Okay, precision. Remind me what that means again.
Precision is about consistency. You want your assay to produce similar results each time you run it, even with slight variations in the experimental conditions. Imagine you're baking cookies. You'd want each batch to come out pretty much the same, right?
Of course, you wouldn't want one batch to be perfectly chewy and the next to be rock hard, even if they all taste good.
Exactly. In science, consistency is key. And MIQE recommends using statistical methods to evaluate the variation in results, ensuring that the assay is producing reliable and consistent data. It's fascinating how much attention to detail goes into ensuring the quality of the data, even before you get to the analysis stage.
It really highlights how scientific depth isn't just about the technical aspects of running an experiment.
Yeah.
But also about the thoughtfulness and rigor applied at every stage.
Absolutely. And speaking of analysis, MIQE doesn't stop at data generation. It guides researchers on how to properly analyze and interpret that data, which is crucial for drawing meaningful conclusions.
So it's like saying, okay, you've gathered all this information now, let's make sure we understand what it actually means.
Exactly. And a key concept here is normalization, which helps account for variations in the amount of starting material and differences in the efficiency of the reverse transcription and PCR steps.
Normalization sounds kind of complicated.
Yeah.
Can you explain it in a way that even I can understand?
Imagine you're comparing the size of two apples, but one was measured with a ruler and the other with a tape measure. You'd need to adjust the measurements to a common scale to make a fair comparison.
Okay.
Normalization in qPCR is similar. It brings the data to a common baseline so you can make accurate comparisons between samples.
Ah, so it's like converting different currencies to make sense of their relative values.
That's a great way to think about it. And the most common approach in qPCR is to use reference genes, which are genes that are assumed to have stable expression levels across different samples.
So you're using these stable genes as a benchmark to measure the changes in your target genes.
Precisely. But MIQE emphasizes that you shouldn't just rely on one reference gene. It's best to use multiple reference genes and validate their stability under the specific experimental conditions.
It's like having multiple witnesses to corroborate a story. It increases the reliability of the account.
Exactly. And another crucial aspect of data analysis highlighted by MIQE is considering biological variation.
So even if you control for all the technical factors, you still need to account for the natural differences that exist between biological samples.
That's right. Even cells from the same tissue can exhibit variations in gene expression. So it's important to use appropriate statistical methods to ensure that the differences you observe are truly significant and not just random fluctuations.
And it sounds like MIQE is really helping researchers to navigate the complex world of data analysis and make sure their interpretations are valid and reliable.
It's like providing a roadmap for researchers to follow, ensuring they reach their destination with the most accurate and meaningful data possible.
So in essence, MIQE is all about boosting that scientific depth, ensuring that every step from experimental design to data analysis is conducted with the utmost rigor and transparency.
Absolutely. And this is not just for the benefit of scientists. It benefits everyone who relies on scientific findings, from doctors making treatment decisions to consumers evaluating new products.
It's really empowering to know that these guidelines exist, helping to elevate the quality and trustworthiness of scientific research that impacts all our lives.
It's like having a quality assurance stamp on scientific findings, giving you that extra layer of confidence in the information you encounter.
Well, this has been an incredibly insightful deep dive into the world of MIQE and scientific depth. It's amazing to see how much effort goes into ensuring the reliability of scientific findings.
It's been a pleasure exploring this with you. Remember, these concepts are applicable beyond just qPCR. Whenever you read about a scientific study, consider the level of scientific depth demonstrated. Are the methods clearly explained? Are the results reproducible? These are questions worth asking to ensure you're basing your understanding on solid science.
Excellent points. So the next time you encounter a scientific claim, think of MIQE and ask yourself, does this research hold up to the standards of scientific depth? If you're curious to learn more about MIQE, check out the links in our show notes, and as always, happy diving.